Zwitter ion. Zwitter ion. See, we can see the anion means the one with a negative charge. We can see the cation, the one with a positive charge. But in some species, both positive and negative charge is present. They are called as Zwitter ions. Or you can write the sentence like this: the molecule in which both positive and negative charge is present is called as Zwitter ion. In amino acids, we can observe both positive and negative charge, which in the same species. Zwitter ions are the di-positive ions. Zwitter ions are having both positive and negative equal charges. Okay, you can write the definition like this: the molecule in which both positive and negative charge is present. The molecule in which okay, now we are doing Zwitter ion only. Now only I started. Okay, both positive, not ion, positive and negative charge. Is called as Zwitter ion. Simple definition. Okay, see all of you. Zwitter ion, the molecule in which both positive charge and negative charge are present, is called as Zwitter ion. Let us take amino acid. In this amino acid group is there, na? That acid group donates proton to nitrogen. Then what we will get? Nitrogen with a positive charge, CH2, and COO negative. This is called as Zwitter ion. Zwitter ion. Amino acids. Which compounds form Zwitter ion? Amino acids. Amino acids form Zwitter ion. Okay. Online students, are you following, all of you? Right. Next, isoelectric point. Next heading, isoelectric point. You can write the definition. I'll send the PDF. Isoelectric point. PI, abbreviated as PI. PI stands for isoelectric point. What is isoelectric point? At particular pH, tell me the pH at which Zwitter ion exists. See, next concept, isoelectric point. At a particular pH, this Zwitter ion do not move towards cathode or anode, is called as isoelectric point. At a particular pH value, Zwitter ion do not move towards cathode or anode. That is called as isoelectric point. For example, if we take amino acid is having a positive charge, and Zwitter ion is having positive charge and a negative charge. So it may move towards cathode or anode. If you do electrolysis, you know electrolysis. What is electrolysis? Passing current. What happens when we pass the current? Negative ion will move towards negative ion will to positive rod. Positive ion will move towards negative rod. Clear? Here positive and negative both are present in Zwitter ion, but at particular pH. Deuterion do not move towards cathode or anode. That point is called as isoelectric point. Clear? So how to define isoelectric point? At a particular pH, zwitterion do not move towards cathode or anode. Clear? Right. At a particular pH, the zwitterion do not move towards cathode or anode. Isoelectric point. PA. Calculating isoelectric point. Calculating isoelectric point. PA. See, I'll give you one formula. Amino acid. We know types of amino acids: acidic amino acids, basic amino acids. In case of acidic amino acid means the amino acid in which more acid groups are present, more COH groups are present. Acidic amino acid. Basic amino acid means more NH2 groups are present. So we have two formulas to calculate isoelectric point in case of acidic amino acid and basic amino acid. In case of acidic amino acid, Pi, isoelectric point Pi is equal to sum of two smallest pKa values. Sum of two smallest pKa values divided by 2. Sum of two smallest pKa values divided by 2. In case of basic amino acids, isoelectric point Pi is equal to sum of two largest pKa values divided by 2. Right. Example. You tell me this is which amino acid? Acidic amino acid. Okay. Why? More COH, more acid groups are present. They will give pKa data. You no need to remember any data. You have to remember the formula which I given to you. Here I am giving pKa values. pKa1 is equal to 4. pKa1 is equal to 2. pKa. Let us give 1, 2, 3 like that. Okay. pKa2. Or we can take pKa also, nothing will happen because we add two smallest pKa values, right? Or you can take like this also, nothing will happen. 9. Clear? Now first observe, this is acidic amino acid. What is the formula to calculate isoelectric point? Sum of two smallest pKa values divided by 2 over. So Pi, isoelectric point, is equal to what are two smallest pKa values? This one and this one. Remember the formula. Means pKa1 plus pKa2 divided by 2. Correct only, na? Smallest pKa values. So 4 plus 2 by 2, that is equal to 3. But you will get like decimals, like some like 4.2, 2.7 exact values. You may not get. Clear? Some addition, do addition carefully. Clear? This is which amino acid? Basic amino acid. In case of basic amino acid, take largest pK values by 2. Isoelectric point Pi is equal to largest means these two only, na? So 10 plus 8 divided by 2, that is equal to 9. Test for amino acids. Test for amino acids. Yesterday I given one code for aromatic amino acids, benzene ring containing. Tell me. Rosie? Rosie did Finland. Very good. Rosie means tyrosine. Tyrosine only now. Tyrosine. Rosie stands for Finland. Finland means phenylalanine. Trip means tryptophan. Tryptophan. This is also very important. Test for aliphatic amino acid, ninhydrin test. Highlight this. Ninhydrin test. Ninhydrin test. Which of the following test is used for the amino acids? Ninhydrin test. Especially for aromatic amino acids, benzene ring containing. Xanthoprotic test. Xanthoprotic test. Test for amino acids. They will give direct question. Which test is used to detect amino acids? Ninhydrin test. For aromatic amino acids. Aromatic amino acids means particularly that code I given in the last class. Tyrosine, phenylalanine and tryptophan. That are give positive test with xanthoprotic. Right. Next. Completed? Again you are writing. I said I'll send the PDF, but okay. Next, peptides. Peptides. Peptides are the polymers of amino acids. Peptides are the polymers of amino acids. Contains amide bond. Contains amide bond. Amide bond, a covalent bond. A covalent bond. See, how peptide is formed. What you asked? Same only. Same only. You can call that that amide bond, newly formed amide bond only peptide bond. Compounds in which amide bond or peptide bond, both are same. I'll tell you with this example, your doubt will be cleared. I am taking two amino acids. One is glycine and one is alanine. What is structure of glycine? Glycine structure. C double bond O. This is, what is its name? Glycine. This is alanine. Alanine. They lose water molecule. Sorry. They lose water molecule. Right, remaining structure H2N CH2 C double bond O NHCH. This amide link is nothing but peptide bond. Amide link is nothing but peptide bond. Peptide. Okay, Adiba. What is peptide? That amide link only peptide bond. Amide link. And very important point. We got peptide from how many amino acids? How many amino acids we took? 2. So this is called a dipeptide. Dipeptide. The peptide which is formed from 2 amino acids is called as dipeptide. And one more important point, you took 2 amino acids, you got only one peptide bond. One peptide bond. Clear? When you take 2 amino acids, we will get one peptide bond. When we take 3 amino acids, for example, if I put another amino acid here, from OH and NH2 water will lose, okay, and there we'll get one peptide bond. So if we take 3 amino acids, 2 peptide bond. If we take 4 amino acids, clear? So if N is the number of amino acids, small n, number of peptide bonds are n minus 1, n minus 1, okay, right. Completed this one. Types of peptides. Types of peptides. Peptides are the polymers of amino acids, right? A simple classification. Dipeptide. Dipeptide, right? Formed from, tell me, 2 amino acids. 2 amino acids. Dipeptide contains how many peptide bonds? 2. Because di is there, na? You will think like that only. But one very important. Dipeptide means di means 2. So we may think like that also. But dipeptide contains only one peptide bond. Very important. Contains one peptide bond. One peptide bond. Right? Tripeptide. 2 are enough because remaining concept is similar, right? I'll give you only 2. Tripeptide formed from 3 amino acids. 3 amino acids. Contains 2 peptide bonds. 2 peptide bonds. Okay, right. Write one note. N is the number of amino acids, then n minus 1 is the number of peptide bonds. Number of peptide bonds. Are you following, online students? Online students, everyone, answer this question. A short peptide, again don't get confusion. Peptide means it is a polymer of amino acid contains peptide bonds. Peptide is a macromolecule formed by amino acids. Clear? Peptide means, just imagine, it is a polymer macromolecule which contains peptide bonds. Don't get confusion. Okay, right. A short peptide upon hydrolysis, see, when we do hydrolysis, peptide bond will break and we get amino acids back. Reverse. Once look at this. We are removing water here, right? Did you write minus H2 over there? We are removing water. What happens if we add water? We'll get amino acids. Reverse. Clear? Hydrolysis of peptide gives individual amino acids. Clear? One question. A short peptide upon hydrolysis give 3 Na glycines, 4 Na alanines, 4 Na alanines. Not single 3. I am taking Na, 3 Na, 3 into Avogadro number of glycines, 4 into Avogadro number of alanines upon hydrolysis. Okay, then number of peptide bonds, in that short peptide is dash Na. You can answer direct. Dash Na. Guess. Look at that, hint is there only. 6 Na. Clear? 6 Na, because 3 plus 4? 7 Na amino acids are present in that. 7 means 6. Minus 1 is the number of peptide bonds. If n is the total number, then n minus 1 is the number of peptide bonds. So answer is 6 Na. 6 into Avogadro number. 6 Na. Next, representation of peptide bond. Representation of peptide bond. Representation of a peptide bond. A representation of a peptide bond. Or peptide bond, not peptide. Actually, correct word is peptide, not bond. Peptide. Representation of a peptide. N terminal, C terminal. N terminal and a C terminal. If we consider this box as a peptide, maybe a dipeptide, maybe tripeptide, any peptide. For example, let us take a dipeptide, which is formed from two amino acids, right? Always N should be in the left hand side, whatever it may be, the peptide, dipeptide, tripeptide, tetrapeptide, anything, N should be on the left hand side. C means COOH, C terminal means COOH, N terminal means NH2, N terminal means NH2, C terminal means COOH. COOH should be present on the right hand side. Clear? NH2 should be present on the left hand side. That is the rule. Representation of peptide. N terminal. My name will come first, no? Remember. I am always first. Okay. See this example. Simple amino acid, glycine, NH2, CH2, C double bond. I am writing directly NH, CH, CH3, COOH. See, this is N terminal and the C terminal. You may get questions on this. I am coming to that only. Amino acid, peptide, protein. Writing chestunnara. Amino acid, peptide, protein. See, peptides are formed from amino acids. Proteins are formed from peptides. See this. Amino acids combined to give peptides. Proteins. You know, in human body proteins are present. Proteins are made up of peptide chains. See, this is one peptide chain, polypeptide chain. This is one polypeptide chain. Clear? And some peptide chains coiled around, you know, this helical structures, everything. Proteins are the polymers made up of peptide chains. I am going to give that only. Proteins are the polymers made up of peptide chains. See, what are the basic units of protein? Fundamental unit, amino acids, basic units. Okay. Classification of proteins. First one heading, fibrous proteins. See, according to textbook's definition, in fibrous proteins, peptide chains run parallelly, nothing but arranged parallelly. Peptide chains arranged parallelly. Run parallel in the sense arranged parallelly. In fibrous proteins, peptide chains run in the sense arranged. In our terminology, arranged, parallel or parallelly. Just for understand, can draw like this. This is one peptide chain, this is one peptide chain. You know how peptide chains are formed? Amino acids combined, they lose water molecule and peptide chains formation takes place. This is polypeptide chain, means actually we know dipeptide, we know tripeptide, we know tetrapeptide, but this is polypeptide. So many amino acids combined to give polypeptide chain. This is one polypeptide chain, this is one polypeptide chain. They run parallelly. Interactions. Hydrogen bonding, intermolecular, and disulfide bonds. Disulfide bonds. How sulfur will come? Sulfur containing amino acids are present or not? Okay. Note down interactions. Interactions also important. Water solubility. Soluble or insoluble? Soluble or insoluble? You know, I'll tell you one simple trick. Hair is soluble or insoluble? Hair is soluble or insoluble in water? That is also doubt only, na? Hair is soluble in water. If hair is soluble in water, we will get bald head. Hair is insoluble. Okay? Insoluble. Insoluble in water. Insoluble in water. Examples are very important. Examples list. Keratin, myosin, collagen, elastin. Keratin is present in, I think, hair and nails. I think it is correct or wrong, I don't know. You only tell me where it is present. Hair or nail? Hair. Myosin, I know about myosin. Myosin, muscles. Collagen? Okay. Next, globular proteins. Globular proteins. Globular proteins. See, in fibrous proteins, polypeptide chains are arranged parallelly. In globular protein, they coiled around to give a spherical shape, spheroid shape. Okay. In this polypeptide chains, coiled around to give spherical shape. These are soluble in water. Soluble in water. Fibrous proteins are insoluble in water. These are soluble in water. Interactions. Hydrogen bonding. Examples list insulin, albumins, hemoglobin. Right. Examples are very important. Examples category. Next, structure of proteins. Structure of proteins. Tell me structure of proteins. Online students. Structure of proteins. Primary structure, secondary, tertiary, quaternary. Right. Structure of proteins. First one, primary structure. Primary structure. What is a primary structure? In primary structure, specific sequence of amino acids is present with peptide bonds. Specific sequence. It is determined by DNA, genetic coding. Right? Note down. In primary structure, DNA test, you know about DNA test. Specific sequence of amino acids with the peptide bonds. With the peptide bonds is present. Primary structure is determined by DNA genetic coding. It is determined by genetic coding. Primary structure is the first level organization of proteins. You know classification of what is that animals are, what is that first level organization in the first chapter, the first year. It stands for primary structure. It is first level organization of proteins. And one important point, primary structure is stabilized by very important point, covalent peptide bonds. Very important point. Primary structure is stabilized by covalent peptide bonds. Strong bonds. We use this point in the denaturation concept. Remember, primary structure is very strong. Primary structure is very strong because of covalent peptide bond. Amide bond is a strong bond. We use this point in the denaturation of proteins concept. JVSR students, you can leave because I'll complete this and I'll send the video to you. Theory only, what is that, zoology background only, this is you can manage. I'll complete the portion. Okay, Meenakshi and Bhakti. I'll mention the timings, you watch from that time. Okay. Next. Tell me, primary structure is stabilized by? Covalent peptide bonds. Covalent peptide bonds. Are you getting, online students? Are you following, all of you? Right. Next, secondary structure. Secondary structure. Right. Primary structure means specific sequence of, in which of the structure specific sequence of amino acids is observed? Primary structure. Secondary structure. I'll give about secondary structure only and properties of tertiary structure. Okay, tertiary and quaternary. Secondary structure. See, secondary structure is formed due to folding of peptide chains due to hydrogen bonding. Due to hydrogen bonding. Because of hydrogen bonding, they fold or twisted because of hydrogen bonding. The structure which arises due to folding of polypeptide chain backbone due to hydrogen bonding. Hydrogen bonding especially between NH and C double bond O. Clear? That is called as secondary structure. Secondary structure is two types: alpha helix and beta pleated sheet structure. Clear? Right. Secondary structure. It arises due to folding of polypeptide chains to hydrogen bonding between NH and C double bond O. Secondary structure is two types. Alpha helix, beta pleated sheet. Two types of structures: alpha helix and beta pleated sheet structures. See, simple definition I will give you. In this, polypeptide chains are twisted. Here, polypeptide chains are stretched. Okay. In alpha helix, polypeptide chains are twisted. Here, polypeptide chains are stretched. In zoology, you learn about briefly, but in this chapter, just basic fundamental information is enough. Primary structure is stabilized by covalent bond. Secondary structure is stabilized by hydrogen bond. Tertiary structure is stabilized by many factors: ionic bonding, hydrogen bonding, van der Waals interactions, disulfide interactions, several. Okay, now don't mention that point. secondary structure is stabilized by H bond, hydrogen bond. Tertiary structure, I'm not giving the information. Actually this is a overall three-dimensional structure of the protein. Okay, or we can write like this: overall 3D structure. Simple sentence: overall 3D structure, because information is very vast. Okay, we have to spend more time to get clarity on them. But for regarding chemistry, we get basic information only. Basic information is sufficient. But here this point is important. 3-degree structure is stabilized by. You can note down that 3-degree structure is stabilized by. Write one by one. Hydrogen bonds, ionic bonds, van der Waals forces, disulfide bonds, okay. Primary structure is stabilized by strong covalent peptide bonds. Secondary and tertiary structures are stabilized by hydrogen bond, ionic bond, van der Waals, disulfide bonds, everything. But all these are very weak compared to that strong covalent bond. Covalent peptide bonds are very strong. Why I am stressing that point? See, in secondary structure and tertiary structure, interactions are present, bonds are present, like hydrogen bonding. That is a weak interaction. Clear? In the primary structure, strong interaction is present. Covalent peptide bond is strong bond, strong interaction. That's why during denaturation of protein, primary structure won't be disturbed. Very, very important. What is denaturation? Denaturation of protein means? Denaturation means you won't get all these things in your zoology. You won't get this. PCA technique. She's saying something. Biotechnology something. Okay, denaturation of protein means, see, changing the property of a protein. For example, egg, albumin, egg white. What happens when we boil? Simple thing, its liquid shape converted to solid. We are changing its property or not? Curdling of milk. In milk, one protein is present. During curdling, it is changing or not? Physically, if we take it, is converting into some solid type substance. So changing the property or biological activity of protein by doing some operations in the sense by adding acid or base, means changing the pH, by heating. Best example, boiling of the egg. Physical method means boiling of the egg, agitation, mixing. All these are very, very important. Which of the following is the denaturation process? Okay, adding urea, adding like metals, heavy metals like Pb plus 2, Hg plus 2. All that are process of denaturations. Methods used for the denaturation. Denaturation nothing but changing the property of the protein or changing the biological activity of the protein by doing all these operations, by heating or by adding some chemicals. Clear? Right. But very, very important point, during denaturation, which structure is do not disturbed? Primary structure or? Primary structure. Why? Because in primary structure, strong covalent peptide bonds are present. During denaturation, secondary and tertiary structures are disturbed. Very, very important point. Because all these are weak forces like hydrogen bonding, van der Waals forces. Clear? Right? Next heading, denaturation of proteins. denaturation of proteins. Changing the structure or biological activity of proteins by subjecting protein to physical or chemical change. Physical or chemical change is called as denaturation of protein. Examples of denaturation methods. First one, all these are important examples. Changing pH of protein means adding acid or base. By changing temperature. By agitation means mixing. By agitation. By adding urea, alcohol. All these are the methods. By adding heavy metal cations, Pb plus 2 or Hg plus 2. During denaturation, what happens? Very, very important point. Protein loses its biological activity and secondary and tertiary structures get disturbed. There is no change in the primary structure. That is only the important point about denaturation. Very important point. Primary structure do not disturbed. Why? Due to strong covalent peptide bonds. Secondary and tertiary structures. My guess questions will come on this. Secondary and tertiary structures will disturbed. And another point, you tell me, another important point, proteins lose their which activity? Proteins lose their biological activity. Proteins lose their biological activity. Can I add this point also? Add this one also. Fibrous proteins do not get disturbed. Fibrous proteins. To hot water bath, head bath, hair won't be disturbed. Keratin comes under fibrous protein. Fibrous proteins is additional. Globule proteins disturbed. Albumin, egg albumin. Egg albumin is the best example or not? Albumin. We can boil the egg. See, put your head in hot water. Nothing will happen to your hair. Your face will burn, but your hair do not disturb. Don't do like that. Okay. Next. Proteins after enzymes. Keep heading enzymes. How food is digesting in our stomach? By chemical reactions. Digestion is a chemical process. Respiration also a chemical process. Okay. For chemical process, some catalyst is required. That are nothing but enzymes. Enzymes are biocatalysts. In our body, human body or animal body, biological reactions takes place. For that reactions, some substances act as a catalyst. That are nothing but enzymes. These are biocatalysts. You tell me, catalyst is required in large amount or not? No, small amount. Catalysts always required in the small amounts. Clear? Just if you have small amount of catalyst, reaction will speed up. Very, very important. Generalization, that is, catalysts are required in a smaller quantity. These are required in smaller quantity. These are required in smaller quantity. And very, very important, you tell me, digestion of food is one chemical reaction. Respiration is one chemical reaction. Clear? Can we send enzyme which is used to digest the food for the respiration process? Cannot. So enzymes are specific. Okay, they are specific, means for a specific biological reaction we use specific enzyme. These are specific for particular substrate and particular reaction. Clear? Enzymes are specific. For particular biological activity, particular enzymes are required. One enzyme cannot act as a catalyst for all biological reactions. Clear? Right. These are specific. For particular substrate. And for particular reaction, biological reaction. What is the role of catalyst? Generally I am asking you, but in kinetics also over catalyst. Catalyst increases or decreases the activation energy? Increases. If activation energy increases, then reaction slow down. Catalyst role, not only biological catalyst, general catalyst decrease the activation energy and speed up the reaction. Clear? When activation energy decreases, reaction will become fast. Once you see the chemical kinetics, physical chapter. These enzymes, these means enzymes, these enzymes decrease activation energy, write properly. Activation energy and speed up the reaction. Right? Theoretical questions also important. Activity or role. enzyme activity or role. Diastase, enzyme diastase. Where you have seen this diastase? Starch to maltose. Starch to maltose. In carbohydrates are given. Which enzyme is used? Diastase. Which one? Diastase only. Under starch.
Glucose to xylase.
Glucose to alcohol. Zymase. That is different. Glucose to alcohol. This is starch to maltose. What is the formula of starch? Starch formula is given. C6H10O5 taken N. C6H10O5 taken N. Maltose C12H22O11. Maltase. Guess maltase. Maltose to glucose. Zymase. Glucose to alcohol. Pepsin. Protein to peptides. Note down this. I'll come. Okay. Hello, online students. Are you there? Next. Sorry. You can leave and see the video. Okay. Next, nucleic acids. I'll post the video. You can watch later. Nucleic acids. Last topic, nucleic acids. DNA and RNA. What is the full form of DNA? Deoxyribonucleic acid. Ribonucleic acid. DNA or RNA when subjected to hydrolysis. Tell me, what are the products of DNA and RNA? Hydrolysis products. Don't know? Pentose sugar plus nitrogenous base and H3PO4, phosphoric acid. DNA or RNA when subjected to hydrolysis will get pentose sugar, nitrogenous base and phosphoric acid, H3PO4. Okay. Pentose. Nucleic acids, DNA, RNA, hydrolysis of DNA, RNA. Clear?